Staying on the topic of CNS, we're going to now look at an ALS company, and I want to ask everybody a question. You've been listening to a lot of presentations today, and it took me a while to figure this out, but what do mesenchymal cells like to do? They like to home. So what if you could engineer a mesenchymal cell that would be a super secretor of BDNF and GDNF, and you could put it in a position where it could home to the sites of injury? I think that's what makes BrainStorm, BCLI, very, very interesting in the ALS space. Very, very novel approach. I've visited the company. I've been to Israel. I've kind of talked to the investigators. This is something we want to pay attention to. Today we have Chen, who is here from BrainStorm, who's a board member of the company, who also is the former head of business development at Teva. And like most people in Israel, everybody worked for Teva at some point in their career. Thank you very much. We're looking forward to hearing about BrainStorm.
Thank you. Thank you for the introduction, and thank you for your interest in BrainStorm. I'm going to describe what we have achieved during the last few years, because the science has progressed very, very significantly. We're now at the point in the company where biology, preclinical, phase 1, phase 2, all the data that we have collected over the years are coming together and provide a very good proof of concept to the approach that we're taking. BrainStorm is developing cell therapy for the treatment of neurodegenerative diseases. We started with ALS. We'll talk in a minute why we selected the ALS as the first indication. We have successfully completed phase 1, 2, and we've announced the data. We're now in the midst of following up on our second phase 2, and we'll talk a little bit about the data that we've seen so far from this study. And why don't we move and talk about how do we work with our cell therapy. So essentially, the first step in our therapy is harvesting MSC cells, mesenchymal stem cells, from the patient. We grow the cells and we differentiate the cells into cells that look more like neuron and basically generate GDNF and BDNF. So when we inject the cells into the patients, we know, and we check the cells before we transplant the cells into the patients, we know that they generate BDNF and GDNF, and these are very important neurotrophic factors for these patients. So this is how the process works in the different steps for the treatment. And what makes us unique compared to many of the companies that you've seen today? So the first, and there are a few companies that do that, but not many, our cells are autologous. So we take it from the patient themselves. There is significantly lower risk for any rejection on the cells. And indeed, in our clinical studies so far, the safety profile of our approach has been very, very promising, and we didn't have any significant issues from a safety perspective. We then take these cells and differentiate them into cells that can actually provide the benefit to the specific disease, ALS in our case, because we check those cells to make sure that they secrete the right neurotrophic factors before we inject them. This is just a picture. A picture is worth 1,000 words. You can see the mesenchymal stem cells here once we harvest them, and you can see that after the process of the differentiation, these cells look much more like neurons, and you can see the GDNF and the BDNF in these cells. So we actually check, and we can see even here some data from patients. So you can see in blue the MSC cells, and you can see in red, following the process, and these are, different patients, you can see how these cells secrete, in this case, BDNF and GDNF. So from a biology perspective, before we transplant the cells, we make sure that they secrete the right factors. This is just some proof of concept in a preclinical model. This is the preclinical model for ALS. What you expect to see is survival, and we see benefit for survival and benefit for the motor function. So moving into the clinical studies. So far, we've done 29 patients in our clinical studies. We did a couple of compassionate use studies, and sometimes from these type of studies, you learn the most about the benefit of the therapy we had cases, and I believe this was announced, or there is a case study that was published. We had cases of patients that came into the study without very good ability to speak. Their own family couldn't really understand what they're saying. They may be in a wheelchair, and after treatment, one could see them actually, I wouldn't say walk, because this is not a miracle, but definitely communicate better, even speaking in front of a crowd. So you can see phenomenal benefit to these type of patients from cell therapy. And these stories continue and continue, and we get many requests from patients that would like to participate in our clinical studies. I'm going to speak a little about our phase 1/2 study, and I'm going to speak a bit about our phase 2A study. There's actually going to be some data that we're going to show today from the phase 2A study. We expect to initiate a phase 2 in the US later this year, and we expect to start a phase 2B later this year in Israel. So let's talk about our phase 1/2. So the way the process works is the following. We recruit the patient, and during the first 3 months, we follow up on the patients. What does follow-up means? We look what is the rate of the decline in the scores that we want to measure in ALS clinical studies. We'll talk about these scores in a second. We then transplant the cells after we differentiate the cells and we grew the cells, and then we monitor what is the rate of the decline over the next period following the transplantation of the cells. In terms of safety, no surprise, safety profile has been very, very promising, and since this is an autologous approach, we didn't really expect any issues from a safety perspective. So let's see the data. So these patients usually live between 2 to 5 years. They die either from complication of paralysis or from respiratory failure. So there's 2 key endpoints in ALS studies. One is the ALS functional rating scales, which is ALS-FRS, and another key endpoint is one that we'll describe in a second. So you see on the left side, you can see the rate of the decline of these patients before they receive treatment. So you can see around negative 1.5. Following the treatment, you can see that the disease, is pretty stable over the next few months. That is very rare to see in ALS clinical studies. We do see that, at some point the disease continues to progress, and we do expect that over time patients will get repeated treatments every 3 to 6 months. So it's probably going to be a therapy that will be administered between 3 to 4 times a year. So what we see in this slide is a decline in the rate of the progression in the ALS-FRS score. This is something that we rarely see in clinical studies in ALS. And then if you remember, I mentioned that one of the key reasons eventually for death of these patients is respiratory failure. Forced vital capacity basically measures the lung function. And again, same story. You can see a pretty, steady decline in those patients before they start the study, and you can see that this decline is slowed to a certain extent following the therapy. Very important benefit to patients. Another area that's important to analyze, and that's something that also regulatory agencies will probably look at over time, what is the change in the different scales? How do patients benefit actually from the treatment? So neurological exam, you can see very significant benefit to these patients. Forced vital capacity, again, very significant patients benefit. And also if you look at some of the measurements of the right arm and left arm muscles, you see benefit to these patients. So let's just, see so we've seen the biology, we've seen the preclinical, we've seen the data of our first phase 1/2 study, and then we're going to talk a bit about our data from our currently ongoing phase 2. We're basically in the stage where we're following up on patients. So this study was again in 12 patients, and we're looking at 3 different doses. In terms of the progress of the study is pretty much the same process. It's a 3 months follow-up before they enter the study. During that period, we harvest the cells, we grow the cells, we differentiate the cells, and then we transplant these cells into the patients. And again, it's very encouraging. These are the data that we have kind of so far. very encouraging. You see the progress of the disease before these patients enter the study. If you look at, overall the progress, that's the ALS-FRS, and you can see that following the injection, it's pretty, it's lower. It seems to slow the disease progression, and the numbers just speak for themselves. If you look at this patient here, you can see a pretty significant decline, and they'll almost steady state. If you look at the blue patient here, again, pretty steady state. In these patients here, again, let's not look at those bumps, but overall pretty steady state. This one, slight decline, but overall seems to slow disease progression. Going to the next slide, forced vital capacity. Second clinical study where we measure the respiratory function. Again, pretty clear. Before, you can see overall a decline. After, I think most of these lines seems to be very flat. So and they're very flat over a period of 4 to 5, maybe 6 months. As I mentioned, I do expect eventually that at the commercial phase, we'll dose patients probably 2 to 3 times a year. We expect to initiate a phase 2 study in 48 patients this year. This study will be conducted in 3 leading centers, and you can see the names of the principal investigators. These investigators are, pretty well known in this country. We expect to announce the initiation of this study in the very near future. We've already announced that we signed agreements with some of these centers, and we expect that to move us closer towards approval eventually in the US. From a milestone perspective, we expect this year to announce the results of our phase 2A. We gave you, some glimpse into what we expect those data to look like. We expect to initiate the phase 2, repeat those study in Israel, and initiate the phase 2 in the US. We also expect some news on the manufacturing front. As you can imagine, in these type of therapies, manufacturing is very important, and we're well in our way to progress how we manufacture and towards commercializing the technology for manufacturing. So in terms of what have we done, what have we shown over the last period, we've shown that the biology works. We've shown it in preclinical that it works. We've shown it in our first phase 1/2 that it seems to slow disease progression on the key scales that are required for regulatory approval. We are hopefully going to show it in our phase 2 that's currently ongoing. Initial data from the phase 2 seem to suggest that it's working in the same way. Thank you.